Hi, welcome. This is the Chapter 11 review for the cell cycle and cell division, and this is part 3 looking at meiosis specifically. So if we take a second to remember where we're at, we had previously learned about mitosis, which is a type of nuclear division followed by cell division, cytokinesis, which is used for growth, repair, and regeneration. And this is the figure at the top. You start with one cell, you go through one round of nuclear division and then cell division. We get two identical daughter cells. Okay, that's great for growth and repair. In humans, when we create our gametes, we use a process called meiosis, and this is for reproduction, and this is the figure on the bottom. And this is where you start with one cell, and you go through two rounds of cell division, and the end result are 4 cells, which are not genetically identical to the cell we started with. They have variation. And one of the biggest differences is that they're haploid. They have half as much genetic information as the cells we started with. Okay, so before we look at meiosis, let's overview some of the key terms that we need. Okay, so here's a cell. This is a typical diploid cell. And in this case, the cell is diploid, so it has 2 copies of all its chromosomes. And this cell has 3 chromosomes. And it has 2 copies. So here's chromosome 1. This is the maternal chromosome, which means the chromosome from this cell got from its mother. Here's the blue copy of chromosome 1. This is the paternal, which means the chromosome this cell got from its dad. So it has one chromosome 1 here, one chromosome 1 here, so it's diploid. Okay, here's chromosome 2. There's 2 copies of chromosome 2, and there's 2 copies of chromosome 3. Now if we zoom in on one of these chromosomes, here's chromosome 3. This is only one copy of chromosome 3, the paternal copy, but it's gone through replication. So here's one copy of it, here's a replicated copy of it, and these are called sister chromatids. Okay, so we call these sister chromatids. Now if you have 2 chromosome 2s, now note here, these are completely separate. They're not joined together. There's a blue version, a red version. This is the one from dad, this is the one from mom. These are called homologous pairs of chromosome. Okay, that's one of the same chromosome from each of the parents. Okay, so sister chromatids are copies of one actual chromosome, chromosome 3 from dad in this case. Homologous copies are 2 separate chromosomes. Okay, 2 of the same number of chromosomes, so chromosome 2 from mom, chromosome 2 from dad, they're a homologous pair. All right, so this chromosome 1 right here and this chromosome 1 right here, these are also homologous pairs. And then this chromosome 3 and this chromosome 3 are homologous pairs. All right, so we need those terms because as we go through meiosis, we're going to keep track of these. Now here's the typical life cycle for humans. And in this figure, we see mitosis and meiosis in terms of how they're used. So here's the adults, right? They're multicellular and they're diploid. And that means they have 2 copies of all their chromosomes. So we have 23 chromosomes, but we have 2 copies of them, so there's 46 total chromosomes. So mom is diploid, dad is diploid. 46 chromosomes in all their body cells. Now what happens when they make their gametes, okay, through this process, meiosis, they take the number 46 and they split it in half. Okay, so the egg has 23 chromosomes, the sperm has 23 chromosomes. Each of the gametes are haploid, so we say the gametes are haploid. This is important because now if you take an egg with 23, a sperm with 23 chromosomes, and you join them together, we create a fertilized egg called a zygote that has 46 chromosomes. So now we're back to that diploid number again. So mom and dad are diploid. They each give 1 copy of all their chromosomes in their gametes. So the gametes are haploid. They have half as many. When the gametes come together, they're diploid, and it has 46 chromosomes. If mom and dad didn't split, mom would give 46 chromosomes, dad would give 46 chromosomes, and when they came together, we'd have 92 chromosomes in the zygote, and that would be way too many, so we don't do that. So meiosis is responsible for creating our gametes. Once they form a zygote, that zygote then develops through mitosis into the adult organism. Okay, so from here it has 46 chromosomes, and every time it divides, as the organism grows, each of those cells keeps 46 chromosomes. If we look at it here, we can actually see the chromosomes. So here's mom and dad, they're diploid. Mom has 2 chromosome 1s. Okay, she has a blue version and a red version. Dad has 2 chromosome 1s, a blue and a red version. Through meiosis, we create gametes that only get 1 of these copies. So by chance, mom's egg has the red copy and dad's gamete has the blue copy. Doesn't matter which copy they get as long as they get one chromosome 1 in each of these gametes. Now when they fuse together to create a diploid zygote, it has 2 chromosome 1s, okay? And now this is a diploid. Now you got to remember, this is happening for every single one of the chromosomes out of the 23 pairs. Also note, chromosomes only look like this after S phase of interphase, and that'll make a little more sense in a minute. Okay, so here's a general overview of where we're going with meiosis. The main idea is we're starting with a diploid cell. So a diploid, so it has 2 chromosome 1. So if this is chromosome 1, it has a red version and a blue version. So that's what we call a homologous pair of chromosomes. Before meiosis I or meiosis II, we have S phase. This is really important. During S phase, this is S phase of interphase, just like we learned about in mitosis, each chromosome makes a copy of itself. So this blue one makes a copy of itself, the red one makes a copy of itself. So now we have sister chromatids, right? And together these make up a homologous pair. And what's really important is S phase only happens once throughout the whole process of meiosis. It only happens once right before meiosis I, and it never happens again. So during S phase, we're copying all the genetic information. Then in meiosis I, homologous pairs of chromosomes separate. So here we have a blue chromosome 1, a red chromosome 1. They're going to separate into their corresponding daughter cells, okay? And so this cell gets one chromosome 1, this one gets the other chromosome 1, okay? So the key here is for meiosis I, homologous pairs are separating. And then in meiosis II, the sister chromatids separate. So this blue, this is one chromosome that's replicated, and when it separates in meiosis II, each of these sister chromatids is going to go to a new cell, and when they do that, they become independent actual chromosomes. So these are sister chromatids, they separate into individual chromosomes. So the key with meiosis II is that we're going to be saying sister chromatids separate, okay? And you can also see we start with 1 cell and we end with 4 cells after 2 rounds of cell division. So they're haploid. We end with haploid cells and we started with diploid cells. Okay. The other thing is we only replicate the chromosomes 1 time. S phase only happens once at the beginning, so we copy everything once, but we divide 2 times. So the result is if you copy everything once, but you split the cells 2 times, we're going to cut the amount of genetic information in half. So that's why we end with haploid cells at the end of meiosis. Okay, so let's look at the 2 phases of meiosis, meiosis I and meiosis II. And essentially, we're going to be using a lot of the same terms we've been using with mitosis. And generally, these concepts mean the same thing. So for meiosis I and meiosis II, there's going to be a prophase, a metaphase, anaphase, and telophase, and cytokinesis. Very similar to what we talked about in mitosis. There was a prophase, a prometaphase, metaphase, anaphase, and telophase. These phases, generally the same broad things are happening. But we're going to fill in some of the specifics. So for meiosis I, we have PMAT, P-M-A-T, prophase, metaphase, anaphase, and telophase. Prophase, during prophase, chromosomes become visible, the nuclear envelope disappears, and the mitotic spindles form. And in this case, homologous chromosomes are going to start to align with each other. Then metaphase I, generally something aligns at the center. In this case, homologous pairs of chromosomes align at the center. Anaphase, something separates. So in anaphase I, homologous chromosomes separate, and they move to opposite sides. The sister chromatids are attached, and then in telophase I, 2 haploid daughter cells form. So for meiosis I, the key here is if you look at all these red terms is homologous chromosomes are the things being moved around during meiosis I, and ultimately they separate into sister chromatids. So if we look at meiosis I, during early prophase, what do we have? The chromosomes become visible. That's what happens during prophase. And then in prophase I, the other thing that happens here are homologous pairs of chromosomes line up with each other. So if you look at this diagram here, here's 2 homologous pairs. This would be like chromosome 1 and chromosome 1 lining up. Chromosome 2 and chromosome 2, chromosome 3 and 3. Those are homologous pairs. Now these homologous pairs that line up during prophase I, something important happens here, and this is called crossing over. So when these homologous pairs join up, they form what's called a tetrad. Okay, and when they do this, they're actually going to swap some information between them. So the red one's at the top, the red homologous chromosome, and the blue one. The red and blue one are going to do this process. If you look right here, they're actually going to swap some information, and they create a new varieties. Okay, this is called crossing over when they do this. Okay, crossing over. Now we have a new version. This is an all red chromosome, now has a little bit of blue on it. This is an all blue one, has a little bit of red on it. So this is one way we create genetic variation in meiosis, is crossing over, and occurs in prophase I. It's one of the important phases to remember in meiosis, prophase I, because crossing over occurs. Crossing over is swapping of information between homologous pairs. The next phase is metaphase I. And metaphase is pretty easy. Stuff aligns at the center of the cell. In this case, since it's meiosis I, its homologous pairs of chromosomes align at the center of this cell. Now, here's another important part. Metaphase I is important because when the homologous pairs align at the center of the cell, they can do so in any order. So on the left, we have the top. This is homologous pair for chromosome 1. Okay? And then this is chromosome 2. And the blue versions are on the left, the red versions are on the right. And that's fine. Over here, we have chromosome 1 at the top, chromosome 2 at the bottom. This case, blue's on the top for chromosome 1 is on the left, and the red one is on the left for chromosome 2. They are aligned differently. But it doesn't matter. All that matters, the end result, is that all the chromosome, there's a chromosome 1 and a chromosome 2 on each side. So when they split, all the gametes get one chromosome 1 and one chromosome 2. Okay, one chromosome 1, one chromosome 2. It doesn't matter. If we look at these, they have one of each. Over here, they have a blue and a red, a blue and a red. Okay. Over here, they're all blue or all red. It doesn't matter. But what you do see is 4 different combinations of gametes that could be produced. And so this is independent assortment in metaphase I. So if we look at genetic variation, during meiosis, we had crossing over in prophase I that swapped some information between chromosomes, created new varieties. Independent assortment creates new combinations of gametes. Now this is why siblings don't look 100% alike. This could be a gamete used to make one of the children, and it has a red chromosome 1 and blue chromosome 2. This could be a gamete to make another child, and this has a red chromosome 1 and a red chromosome 2. So they're slightly different, okay? And this helps create genetic variation within families. And humans have 23 chromosomes, so there's over 8 million ways all of our 23 chromosomes could align right here in the middle. We're only seeing chromosome 1 and 2 here, but imagine 23 lined up, all the combinations you could have. Okay, so we're moving along with meiosis, and we've gone through metaphase where homologous pairs line up at the center, right? Now anaphase, things are going to separate, and since it's meiosis I, homologous chromosomes separate. Okay, so now we have homologous chromosomes have separated. Now we just have the sister chromatids. Okay. Okay, that's what happens during anaphase. And then in telophase, the nuclear envelope reforms and the chromosomes relax and you don't see them anymore. And essentially that's the end of meiosis I. We started with one parent cell that's diploid, has 2 copies of all its chromosomes. We went through prophase I, crossing over occurred, homologous pairs aligned with each other. Metaphase I, the homologous pairs lined up. Anaphase I, homologous pairs separated. And we ended here, telophase. They're going to split into 2 daughter cells. And we have genetic variation. If you zoom in, if you could look at these chromosomes, they're not all identical to the ones we started with. There's red ones that have a little bit of blue of them. There's blue ones that have a little bit of red in them. So there's some variation. Okay. They also only have 1 copy of each chromosome. So this has 1 chromosome 1 that's replicated, 1 chromosome 2 that's replicated, 1 chromosome 3 that's replicated. And that's going to be significant. Now in meiosis II, we're going to take these 2 cells and split them into 4 daughter cells. Okay, so meiosis II, again, there's PMAT, prophase II, metaphase II, anaphase II, telophase II, and cytokinesis. And again, in each of these phases, a similar sort of things are going to happen. But the main thing with meiosis II is we're going to be primarily talking about sister chromatids. So here's a list of the steps for meiosis II. Let's walk through each of them. The main idea here for meiosis II is that we're going to be talking about sister chromatids. So prophase, what happens during prophase II? The chromosomes start to condense, they become visible, the nuclear envelope will disappear, the mitotic spindle will form. And the sister chromatids will start to move towards the center. When they get to the center, that's metaphase II. We know metaphase II is when things align at the center. In this case, we have sister chromatids. So there's a sister chromatid aligned at the center. We don't have homologous chromosomes anymore, there's sister chromatids. And so in anaphase II, what's going to separate? The sister chromatids. They're going to go to each side, or the poles of the cells. Okay? Then we have telophase II. This is when the sister chromatids, once they separate, they're now independent chromosomes. They've made it to the other side of this cell. These cells then split through cytokinesis, and we have 4 daughter cells. Okay, so let's look at what we started with. Here's a diploid cell. It's 2N. It's diploid. It has, here's 1 chromosome 1, 1 chromosome 1, 1 chromosome 2, 1 chromosome 2, okay, 3, 3. Now let's look at the daughter cells. It only has 1 chromosome 1, 1 chromosome 2, 1 chromosome 3. So these are all haploid. We've cut the number of chromosomes in half. Each one of these only has 1 copy of each chromosome. Okay, the one we started with had 2 copies of each of the chromosomes, diploid, haploid. So now these are the gametes which can be used to create offspring. The other thing is, genetically, the information is cut in half, but they're not identical. So this blue chromosome has a little bit of red on it. This red one has a little bit of blue. So we've swapped some information between our homologous pairs. You can see that happening. That happens during prophase I of meiosis. Okay, the best way to learn meiosis is to write it out. So I really recommend if you could access this slide from my website, print it out and draw it in to see how things are separated during meiosis. And label everything. Label sister chromatids, label homologous pairs. All right, so if this is a cell before meiosis, it's diploid. Here's 1 chromosome 1, here's the other chromosome 1. Here's chromosome 2, here's the other chromosome 2. It has 2 copies. Now what happens during S phase is you make a copy of everything. So this chromosome 1 replicates, this one replicates. Okay, now the chromosome 2 replicates. Here, okay, and here's the other chromosome 2. Now, through meiosis I, now here's the key. S phase only happens once. We only copy everything once, and then we go through 2 rounds of cell division. So the end result is we cut everything in half. You duplicate everything once, and then you divide it 2 times, and we get half as much at the end. So what happens during meiosis I? Homologous pairs of chromosomes separate. So these red chromosomes here are homologous. These are both chromosome 1. These are both, the blue are chromosome 2. They're homologous. So the result are, we get 2 cells with just 1 replicated copy of each chromosome. Okay, so homologous pairs separate during meiosis I. So we have meiosis I, we have prophase I, metaphase I, anaphase I, telophase I. And during those processes, the homologous chromosomes are lining up and then separating into daughter cells. So now we don't have homologous chromosomes, we have sister chromatids. Okay, this is 1 chromosome that's replicated. This is chromosome 1, this is chromosome 2. Okay, and these are sister chromatids now. Now when we go through meiosis II, what's going to happen? These are going to separate into the daughter cells. What are these? The sister chromatids. So the end result here is we get, each of these cells gets 1 of the chromosomes. Okay, it gets 1 chromosome 1, and it gets 1 chromosome 2. Okay, so these are our daughter cells. During meiosis I, homologous pairs separated into the individual cells. During meiosis II, the sister chromatids separated. Okay, each one goes to a different cell. Now let's look at the daughter cells we started with and the cell we started with originally. The original cell we started with is diploid, or 2N, has 2 copies of everything. 1 chromosome 1 here, here's the other one. Chromosome 2, the other chromosome 2. Here's the daughter cells. They're haploid. They only have 1 chromosome 1 and 1 chromosome 2. 1 chromosome 1, 1 chromosome 2. And we created 4 of them. Right? So we've cut the number in half. So these are our gametes. The only thing doing this, I didn't show you crossing over occurring, but that would have happened during meiosis I. So I really recommend working through this a few times because it really helps you understand it. And write out the terms sister chromatids, homologous chromosomes, and it'll really help it make sense. Now, last thing I want to do here is look at mitosis versus meiosis. Okay, and mitosis we know generally is for reproduction, or for regrowth, repair, and regeneration. Meiosis is for reproduction, right, in humans. But there's other differences. Okay, one thing is mitosis, there's only 1 round of cell division. You start with 1 cell and you end with 2 identical cells. Really important, because if you start with 1 skin cell, you want 2 skin cells that are identical to it, right? But in meiosis, what we do is start with 1 cell, we end with 4 cells, and we go through 2 rounds of cell division, and all the daughter cells are genetically different. And that means they have half as much genetic information as the parent cell they started with, and it's been rearranged through independent assortment and crossing over. Now, if we look at mitosis, what happens during prophase and metaphase, Individual chromosomes replicated. Individual chromosomes, the sister chromatids, line at the metaphase plate right here. Okay, so there's chromosome 1, chromosome 1. They line above each other, all in a straight line. Okay, chromosome 1, chromosome 1, chromosome 2, chromosome 2. All right. Over here in meiosis, what happens, homologous pairs group up with each other. Okay, and they form pairs of homologous chromosomes. And when they line side by side. So you have a chromosome 1 on one side, the other one on the other side. They're joined side by side. In mitosis, they all line in a straight line above each other. So the result is, when these homologous pairs separate in meiosis, each of the new daughter cells is only going to get one chromosome 1 is going to go this way, the other chromosome 1 is going to go to the other cell. Chromosome 2, one's going to go this way, one's going to go this way. Chromosome 3, one goes this way, one goes this way. So the result is, at the end of meiosis 1, each of the daughter cells only has one of chromosome 1, 2, and 3. Okay, the other cell gets one chromosome 1, one chromosome 2, one chromosome 3. But in mitosis, they all line above each other, so each cell is going to get one chromosome 1, another chromosome 1. It's going to get a chromosome 2, another chromosome 2, chromosome 3, and another chromosome 3. And so the result is, the daughter cells remain diploid. They each have 2 copies. But in meiosis, they line up side by side, so each cell just gets one of each of the copies of chromosomes. Okay. So one of the best ways to understand meiosis is to draw it out and label everything and keep track of the chromosomes. But hopefully if you work through this review, it'll help you understand it a little bit better.